Hi, Year 11, and welcome to the third paper of the Biology Paper 1 Practice series based on the advanced information from AQA for the 2022 exam series. So we'll get straight on. Our first question is about cell division and stem cells. Question 1 asks us to define what a tissue is. So a tissue is a collection of cells, so it's definitely not a group of organs with one function. That would be a system. Cells with a similar structure and function. This looks promising. I think it's probably this one. And the organ systems in an organism. So no, it's definitely not that. We were correct. So a tissue is cells with a similar structure and function. Over here, what is the name of the tissue at the growing tip of a plant's shoot? So whenever a plant's growing, you have these stem cells right at the tip that can grow and become any type of cell in the plant, and these are known as meristems. C. Plant cells divide by mitosis so that the plant can grow. Give one other reason why plant cells divide by mitosis. Don't refer to growth. So we've already mentioned growth, so we can't talk about that again, but I do know that the whole point of mitosis is for growth and repair. So if they've already mentioned growth, that leaves us with repair. So it's to repair damaged tissue or replace damaged cells. So if you remember, mitosis is for growth and repair. You can't really go wrong there. Next part looks at another simplified version of the cell cycle. Again, we've got this cell growth here where we're going to have that replication of DNA and more organelles growing before you move on to mitosis, which is where the nucleus divides, and finally you'll end up with the cells dividing themselves. So which two processes happen in this section, this cell growth section of the cell cycle? And chromosomes are copied. Yeah, I think it's going to be that one. Chromosomes separate, not yet. That happens in mitosis. Cytoplasm divides by 2. That happens after mitosis. The nucleus divides. Again, that is mitosis, but the organelles increase in number. So this one, and I think this one. So replication of DNA, more cell structures being produced. And again, these do happen, but they happen later in the cycle. So still continuing question 1, we're looking at comparing the two types of cell division we need to know about. We've got mitosis on the left, which is for growth and repair, and meiosis on the right, which produces gametes. And we've got a nice diagram. It shows us the DNA in each of the cells, and it shows us how many cells are produced, and it's asking us to use this information to compare the two types of cells. So straight away, for mitosis, we produce 2 instead of 4 cells. If you have a look here, so we're only producing 2, whereas in meiosis there are 4. What other differences do we have? So our cells here contain 4 chromosomes, whereas the ones produced by meiosis only contain 2. So cells produced, and I'm being quite precise about how I'm saying this, cells produced by mitosis contain 4 chromosomes. Those produced by meiosis have 2, so they've got half the number of chromosomes compared to the mitosis, the ones produced by mitosis. So you could say that these are diploids because they have the same number of chromosomes as the parent cell, whereas these ones are haploids. And again, I think we've covered all the points we need to here. Perhaps we could say that the cells produced by mitosis are genetically identical to parents. That's not the case for meiosis. And yeah, I think that should do us. Next, a scientist investigated cell division in the growth of the tip of a plant shoot. The scientist recorded data at different distances from the tip of the shoot, and it shows us what we've got below. So when we're very close to the tip, we've got quite small cells. Their mean length is only 22 micrometers, whereas when you're further away from the tip, it's 116 micrometers. So that's considerably longer than these ones. Also, at the tip, 13% of the cells were dividing, whereas if you get further away, you've got no cells dividing. So let's look at the mean cell length first. So let's say the further you are from the tip, the longer the cells. And let's talk about the further you are away from the tip, the fewer cells divide. Tip, or the lower the percentage of dividing cells. Oh, dividing. You could probably also talk about the fact that there is no cell division beyond 30 millimeters from the tip, and that should cover everything. Okay. So question 2 looks at the digestive system again, and the first one asks us, what is the name of the enzyme that digests starch? Amylase is the one that we've seen previously, so we'll use that. And where are most food molecules absorbed? So that's got to be the small intestine. So that's where pretty much all of our nutrients are taken into the body. Figure 1 shows 2 villi. So these villi are kind of the long and thin structures that form the wall of your small intestines. And if you look at a zoomed in picture of a single one of these cells, you'll see that the cells themselves are kind of ciliated. They have microvilli, so they're incredibly small. They've also got lots of mitochondria, which will be useful for carrying out active transport. These images were taken using an electron microscope, and it says give one advantage of using an electron microscope compared with a light microscope. Well, the magnification is going to be higher, that's the obvious one. So you've got a greater magnification. You will also have a higher resolving power. So you'll be able to see things in greater detail. D, what blood vessel was labelled X? We'll have to go back and have a look. So these blood vessels here are really, really thin. They connect arteries to veins, so they've got to be capillaries. Next, it gives us the real length of one villus as 0.8 millimeters, and we need to find out what the image length or the image size will be if we use a magnification of 20. And we've got this equation here, which is quite helpful. So essentially what we've got, if you like to use triangles, so magnification, image, real, I would like to know image, so all I need to do, magnification times real. So image is m times r. So the magnification was 20. You'll know it's a magnification because they put a little multiplication symbol at the start. So it's times 20, for example. So 20 times by the real size, which is 0.8. So 20 times 0.8 gives us 16 millimeters. Even though we've written the correct answer here, I'm still going to write it underneath, underline it, to show the examiner that that's what I want them to mark. Next, we've got 2 cells from the surface of a villus. There are sugar molecules inside and next to each cell. So if we just look at cell A, the first one, there are lots of particles outside and not very many in. So I'm going to label this as a high concentration on the outside and a low concentration inside. And I've got the opposite for cell B. I've got a low concentration outside and I've got a high concentration inside. Cell A, the sugar's moving in. It's going from a region of high concentration to low concentration, and I know that that's got to be diffusion. So the process by which sugar moves into cell A is diffusion. So again, that passive process of diffusion where it's moving with the concentration gradient. So yeah, confident it's that. Name the process by which sugar moves into cell B. So here we have low outside, high inside. So this time we're still moving into the cell, but because we're going against the concentration gradient, we're going to need to do something. We're going to need energy. So the only process that moves things into this sort of cell will be active transport. And again, that's why we saw those mitochondria in the microvilli cells. So you need energy for active transport. That's provided by the reaction respiration, which needs oxygen and takes place in the mitochondria. H, give one use of sugar in the body. So, well, you'd use sugar for respiration. That's the most obvious one. You can also use it to store as glycogen. And you can also use it to make amino acids or proteins. Next, so we've got another copy of the diagram. It's exactly the same as before, but we're going to have to explain how things are adapted to maximize the absorption of sugar molecules. And there's probably 3, maybe 4 things we can say here. So each of the cells have those microvilli. You could also say they're long and thin. This increases surface area. And if you have a high surface area, you have a high rate of absorption. So the wall of the villi is only 1 cell thick. So this is a villus, these are villi, and this wall is very, very thin. It's literally only 1 cell thick. This gives us a short diffusion pathway. This means that any substances that pass through these cells don't have a large distance to travel. Short diffusion pathway. And finally, or penultimately actually, we've got a good blood supply, because we've got lots of those capillaries. That means the blood is constantly being refreshed. So your glucose might move from the food into the blood, and that would cause the concentration of glucose in this blood to increase, which would reduce the concentration gradient. So if you've constantly got that blood flowing, you're always going to have a high concentration gradient. And one more thing we could talk about is the fact that the cells have got lots of mitochondria. This is where respiration takes place. Which provides that energy for active transport. Question 3 looks at communicable diseases, in particular bacteria and viruses. So here we have bacteria cells are different from animal cells. What structure is found in bacterial cells and not in animal cells? Cell membrane, I know that all cells have a membrane. I know that you will find mitochondria in animal cells, but not bacterial cells. So it's definitely not going to be that. Nucleus you'll find in all eukaryotic cells. So not that, so it must be a plasmid. So rather than having nuclei, bacteria cells have either free floating DNA or their DNA is kind of in a ring structure called a plasmid. And which scientific process uses part of bacteria cells? Well, again, the process of genetic engineering will use that plasmid as to transfer genetic information between organisms. So I know it's genetic engineering here. Next we have a table listing 4 communicable diseases, which diseases are caused by bacteria and which are viruses. So measles, definitely a virus. Gonorrhea is a bacteria. AIDS, virus. Salmonella, bacteria. So again, just remember these, we just have to know them, unfortunately. Virus causes measles, give us 3 symptoms. So if you've ever had it, the most obvious thing would be your rash or your red spots. You'll also get a fever, so you'll have a high temperature. You also might notice coughing and sneezing. Bacteria can be killed with antibiotics, give one problem linked to the overuse. So if we use them too much, the bacteria are likely to evolve what's called antibiotic resistance. So this means that antibiotics will no longer work on these diseases, so we'll have no way of treating them. Next one looks at a fungal infection called athlete's foot. So this is an example of a fungal disease in humans. It infects the skin and it grows in moist, warm conditions. So how are you likely to be able to transmit it? So a person with athlete's foot they just walk somewhere with bare feet, probably somewhere damp. And then somebody else comes along, walks on the same area, and they get the bacterial disease. So the skin of another person comes into contact with the same surface. They pick up that fungal disease and they suffer from it themselves. And last thing here, bacteria and viruses can enter the body through the nose and the mouth. Describe how mucus, so basically snot, and cilia, so kind of hairy hair in the trachea and nose, prevent most of these pathogens from reaching the lungs. So mucus, your snot essentially traps the bacteria, the pathogens, and then your cilia move the mucus away from the lungs. This is why if you've got a cold or a cough, you'll find that you've got lots and lots of phlegm, lots and lots of mucus, because loads of pathogens have been trapped and your body needs to get rid of it, and you coughing it up or coughing it up and swallowing it is you getting it away from your lungs as best you can. Question 4 is about photosynthesis, and here it asks us what the correct balanced equation for photosynthesis is. So I'm looking for carbon dioxide, water, going to glucose plus oxygen, and there'll be a 6 in front of the carbon dioxide, the water, and the oxygen. So that's definitely wrong. That starts with glucose. This one starts with oxygen. That's definitely wrong. CO2, H2O goes to C6H12O6. Pretty sure it's this one, but let's check the last. Yeah, that's a load of rubbish as well. So I'm confident that this is the correct equation. So 6CO2 plus 6H2O goes to C6H12O6 and 6CO2. B, what type of reaction is photosynthesis? So photosynthesis takes in light energy. So it's taking in energy from the surroundings, and that must be an endothermic reaction. So photosynthesis, endothermic reaction, remember that. We're looking at an investigation here, so they're changing the effect of light intensity on the rate of photosynthesis. This is looking good because they've used an LED light source which doesn't emit very much heat. That's controlling temperature, and they've also used a set concentration of sodium hydrogen carbonate that will make sure that you've controlled the concentration of carbon dioxide in the water. So good start so far. And you place the pondweed 5 centimeters, measure the rate of photosynthesis by counting the bubbles per minute. Repeat it with different distances. That will decrease or increase the light intensity. But they've said that counting the number of bubbles produced in 1 minute is not entirely accurate. So how could we do this better? Well, the best way would be to measure the volume of gas produced accurately. Best way would be to use a gas syringe. That will give you an actual volume rather than just counting number of bubbles, because bubbles can be different sizes. Or even an upturned measuring cylinder. Again, that would also allow you to measure the actual volume of gas produced. We've only used a minute here, so this is quite a short period of time. So I think a longer period of time would be sensible. So 2 or 3 minutes. Next, it mentions this LED light source, the fact that it does not get hot. This is very important because if we increase the temperature, that's going to increase the rate of photosynthesis. So temperature affects rate of photosynthesis. So if we change temperature, we're not controlling all of our control variables, so we won't have a valid investigation. Also, if it gets too hot, so the enzymes that control photosynthesis will become denatured. So the enzymes controlling photosynthesis, so the reaction would just stop. Next one, the student's result is shown in the table below. Predict how many bubbles of gas were produced in 1 minute if the pondweed was placed 40 centimeters. Okay, and we can see here every time we double the distance, we actually decrease the number of bubbles by a factor of 4. So if I go from 20 to 40, if I double that distance again, I've got to divide 8 by 2, so that gives me 2 bubbles in 1 minute, and that will probably be because there's very little light and very little energy for photosynthesis to occur. could just say reduce by a factor of 4 to be complete. Next, we're going to describe how you could change the method to investigate the effect of carbon dioxide concentration. So we need to know how to change the variable and how to keep everything else the same. So we could vary the concentration of carbon dioxide by using different amounts of sodium hydrogen carbonate. You'll remember I mentioned earlier that that's what controls carbon dioxide concentration, and things that we could keep constant. The most important one for me would be temperature. Of the solution, because as we mentioned earlier, temperature is a limiting factor of photosynthesis, and I'd also keep the distance from the light source constant as well, and that would control the light intensity. Question 5 again looks at photosynthesis, this time more focused on the required practical aspects. Firstly, we need to complete the word equation, so just as we saw previously, it's carbon dioxide that combines with water to give us oxygen and glucose. We saw the balanced symbol equation previously, this is a little bit easier. Light intensity affects the rate of photosynthesis, so we've got our lamp, we have our water, pondweed, and counting bubbles of oxygen there, and it's asking us to describe a method that we could use to investigate that light intensity. So first thing we would do is measure the distance between a lamp and the pondweed. I'm just going to do this in bullet points, but I'd recommend you do this in continuous prose. So I would then count the number of bubbles produced, or the bubbles of oxygen, in a particular time, so probably 1 minute. So count bubbles of oxygen produced in 1 minute. Next thing to do would be to change that distance, and this would vary my light intensity. So change distance to lamp. This changes light intensity. I would make sure that the temperature is controlled, so controlled temperature. Now if I had an LED, that would be best because that doesn't emit heat energy, so I would be fine there. But because I'm stuck using this lamp, I think I'm going to have to use something like a heat screen or a water bath to stop this increasing in temperature. So let's just use a heat screen, that's the obvious one. So just a glass divider here that allows the light energy through but would reflect heat energy. So use that heat screen. Obviously we'd use the same size pondweed, we'd use the same kinds. We could control CO2 concentration using sodium hydrogen carbonate. I would repeat each test a further 2 times, so I've got 3 results. So repeat until you get 3 sets of results. I'd calculate a mean discarding any anomalies. And I think that's everything, yeah. So if you mention all of these points, that should get you 6 marks. So measuring the distance, counting the number of bubbles in a minute to measure the rate of photosynthesis, talk about how you're changing the light intensity, talk about how you're controlling at least 2 variables, so temperature using a heat screen, CO2 concentration using sodium hydrogen carbonate, the fact that you're repeating it, taking a mean and discarding anomalies. These 2 here will increase reliability. This makes sure it's valid. Next part of the question talks about algal cells carrying out photosynthesis. You change light intensity and see what happens to the number of new algal cells that are produced. So this tells us the number of extra cells. So when there's no light, you get no extra cells. You don't have any more cells growing. And all of these values are given in standard form. So for example, 1.65 times 10 to the 6th, that's 1,650,000, so that's quite a lot. It says calculate the total number of algal cells after 2 days when the light intensity was, that's the one we just mentioned, so this one here. So we need to add the extra cells to the cells we had to start with. So let's convert this to normal form. So 1.65 times 10 to the 6 is the same as saying 1,650,000. And so essentially all we're doing is we're making sure that this decimal point moves 6 points to the right. We could have converted this into standard form, so this would be 2 times 10 to the 1, 2, 3, 4, 5. But I've chosen to do it this way, and I'm going to add the 200,000. I mean, you could have just typed it straight into the calculator like this, should you want, and it will give you the same answer. So 1.85 million. So 101,850,000 algal cells. Next, we've got a graph. So we need to use these data to plot this graph, and I imagine it's going to ask us to look at any trends. So first point, no, when the light intensity is 0, there are no extra cells. So 0, 0, already plotted that one. 250, already plotted, so that's done, that's done. 1.65 for the next, so 1.65 will be here. So it's a little bit tricky, but I think we've got every 2 squares representing 0.1. So for 2.15 will be 750, so 2.15 takes us there. 1000 is 2.4, which I think will be there. 1250, 2.5, and it's the same here. So I would strongly recommend that when you're plotting these, again, you will use crosses, otherwise you might lose the mark, and that you use a pencil in case you do make a mistake. And then we want a smooth curve of best fit that goes through as many points as possible in a smooth curve rather than a dot to dot. Give 2 conclusions from the results. Well, obviously, if we increase light intensity, we increase the number of algal cells. So as one goes up, so does the other. Increasing the light intensity increases the number of algal cells. But it's not quite as straightforward as that. We've got something happening at around 1200 to 1200, yeah, 1250 lux. Beyond this point here, if we increase the light intensity, there's no increase in the number of algal cells. So we've reached the top of our graph, so we can use the word plateaus here. So we could say beyond 1250 lux, so increased plateaus. (1250 lux). The number of algal cells plateaus. Last part for this question, explain how an increase in temperature from 20 to 25 degrees would affect the number of algal cells. So increasing temperature increases photosynthesis. Increases rate of photosynthesis. So you have more algal cells. And again, don't forget it's explained, so we need to give a reason for our observation. So here's our reason, here's our observation. Question 6 looks at microscopy again. So here we've got a root hair cell viewed by a microscope. It tells us we've got a magnification of 50 and the image size is 43 millimeters, and we want to know the size of the real size of the root hair in millimeters. So magnification is image divided by real. So if you wanted to use a triangle, this is the one you'd use. So we're working out the real length, which will give us image divided by magnification. So real equals image over magnification. The image was 43 millimeters. The magnification was 50. That gives us a 0.86 as the size of the real length of the root hair in millimeters. Now it's asking for micrometers, so if we're converting from millimeters to micrometers, you would also from micrometers to millimeters, you'd divide by 1,000. If you're going the other way, you times by 1,000. We want to convert millimeters to micrometers, so I have to times that one by 1,000. Gives us 860 micrometers. So 860 micrometers. Next we've got a microscope with an eyepiece lens with a magnification of ×5, and we're trying to use the microscope to observe a slide, and you have a magnification of 50. It's worth remembering that your eyepiece magnification times your objective magnification gives you your total. So straight away I'm going to have to use an eyepiece magnification of 10 to give me a total magnification of 50. So let's see what else we need. So first thing you do is place your slide on the stage. You will start with the lowest power objective lens. This allows you to find what it is you want to look at. So it's easier to find things on a lower magnification. So move your slide as close to the lens as possible without touching it. and then you'll use the fine adjustment or the focusing knob to move it away until it comes into focus. Use focus adjustment knob. To slide away from the lens until it comes into focus, and use the fine focus to get a clear image. And we want 50 magnification, so as we calculated earlier, 5 times 10 would be 50, so we need to switch to times 10 objective lens. So all the stuff we mentioned here is pretty straightforward. We've seen it all loads before, but it's just quite complicated getting it into one big paragraph. Next, we've got another investigation here. So we're looking at how root hair cells absorb water and minerals from the soil. With oxygen, you can see that as time went by, loads and loads of nitrate ions kept on being pulled into or absorbed by the plant, whereas without oxygen, when we got to about 130, it plateaued, it stopped being pulled through. So this suggests to me that with oxygen, you're going to have active transport, and the plant can use energy to pull all those nitrate ions in. Without any oxygen, there's no active transport, so the only way that they can get into the cells is diffusion. But once they reach a certain concentration inside the cell, that diffusion can't happen because there's no concentration gradient. Let's see what they're asking us. Describe the changes in the rate of absorption for seedlings with no oxygen. So what have we got? So the rate was fastest in the first half hour. First 30 minutes. Again, straight away, look, really quick initial rate from 0 to 60, not too dissimilar from when oxygen was present. The rate gradually decreases. And we said it plateaued after about 3 hours. So you don't pour any more in. Explain what the results in the table above show how nitrate ions are absorbed. So more nitrate ions are absorbed in the presence of oxygen. So this tells me that active transport must be used. So definitely got active transport happening, which needs requiring that energy from respiration, which needs oxygen to take place. But some ions are absorbed by diffusion, because even without oxygen we still had some ions absorbed. Nitrate ions are essentials for plants to grow. Describe how nitrate ions are used in a plant to help it grow. The nitrates join with glucose to form amino acids, and these are used to manufacture proteins. synthesize proteins. Question 7 looks at food tests and digestion. So the first question asks us to describe how a student could test for lipids. So these are basically fats and what the positive result would be. So the best one to use would be Sudan 3 preparation, and the positive result would be an immiscible red layer. If it's a solution, that will be floating on top of the solution. If you're rubbing it on a solid, then you'll just have like a red layer just sat on top of it. Next, a high concentration of cholesterol in the blood has been linked with coronary heart disease. Name the type of drug used to reduce this concentration of cholesterol. So these are statins. They tell your liver to stop producing cholesterol, hence reducing the amount in your bloodstream. A new drug has been trialled to reduce the concentration of cholesterol. People are either given the drug or a placebo, and one of the risks is it could cause diabetes, and here are our results. It's asking us to calculate the difference between the percentage of patients developing diabetes in group 1 compared to group 2. We have to give it to 3 sig figs. So we'll need to work out the percentage of each one first. And group 1, we're always doing a little number divided by a big number times 100 for a percentage. So 636 over 12,562, all multiplied by 100. That gives us 5.06288. I'm going to keep this number in my calculator for the comparison in a second. And then group 2, I've got 606 over 12,541, all times 100 again. 606 over 12,541 times 100 gives me 4.83215%. And again, I'm recording this to too much precision, and I'll round at the end. And now I need to know the difference between these two numbers. So that gives me 5.06289 minus 4.83215, and the difference is 0.231%. On my calculator it's 23074, but it's asking for 3 significant figures, so these are our 3 sig figs, so that must round to 1. And as always, write the answer you want marking in the appropriate box so the examiner can find it. We have that new chronic heart disease drug, and we want to explain how we could use that drug to reduce the risk of chronic heart disease. So if you have a lower cholesterol, you have a slower or a lower build-up of fatty plaques in the coronary arteries. So you have less narrowing of the arteries, which is what causes coronary heart disease. So there is less reduction in the blood flow. To the heart muscle. and this prevents the reduction of the oxygen supply. Which in turn avoids heart attacks, which is good. And finally, 3 things that doctors will consider when they're planning to use a new drug. So the first thing they'll look at is toxicity or side effects. Is it going to actually hurt you if you take this drug? Then they'll look at something called efficacy. Does it actually work? Is it effective? Then they'll talk about interactions with other drugs. If you're taking other drugs, are they going to cause problems when they're put in together? You'll need to think about the patient's age, their weight, their health, their medical history, whether or not they're pregnant, the dosage, loads of different things, but these 3 will do us for now.